in this screencast, we are going to talk about enzyme inhibitors and also some enzyme regulatory control. Now, an enzyme inhibitor is any substance that can decrease the rate of an enzyme catalyzed reaction. So, a substance that can decrease the rate of an enzyme catalyzed reaction. Now, an understanding of enzyme inhibition is really important for a few reasons. First, the characteristic function of many poisons and some medicines is to inhibit one or more enzymes and to decrease the rates of the reactions they catalyze. And then second, some substances normally found in cells inhibit specific enzyme catalyzed reactions and thereby provide a means for the internal regulation of cellular metabolism. Now, enzyme inhibitors are classified into two categories, which we're going to talk about more in depth in the next few slides, reversible and irreversible, and those are categorized on the basis of how they behave at the molecular level. So when we talk about irreversible inhibitors, an irreversible inhibitor forms a covalent bond with a specific functional group of the enzyme, and as a result of that renders the enzyme inactive. So irreversible inhibitors, right, covalently bond with a specific functional group of the enzyme and make it inactive, right? So a number of very deadly poisons act as irreversible inhibitors. So examples of this include poisons, toxic metals, antibiotics. So not all things are bad here. We've got antibiotics like penicillin and sulfa drugs. Right, so those are just some examples as far as things that act as irreversible inhibitors. Now, excuse me. Now, the cyanide ion is an example of an irreversible inhibitor. So we're going to look at that specific example. And you can see the cyanide ion right there. Now, it's extremely toxic and it acts very rapidly. The cyanide ion interferes with the operation of an iron-containing enzyme called cytochrome oxidase, and the ability of cells to use oxygen depends on the action of cytochrome oxidase. So, the ability of cells to use oxygen depends on the action of cytochrome oxidase. Right? So when the cyanide ion here reacts with the iron in the enzyme, okay, it forms a very stable complex, which is this complex here. And that means once this is formed here, the enzyme can no longer function. And so that means that then oxygen can't go through the system, right? And so what happens then is that means cell respiration stops. and that causes death within a matter of minutes. Now, an antidote for cyanide poisoning needs to be administered really quickly, and one antidote is sodium thiosulfate. Now, when we look at this, what happens is when you have thiosulfate, instead of reacting with cytochrome oxidase, the cyanide ion will react with the thiosulfate and form thiocyanate, and that does not bind to cytochrome oxidase. But this is the cyanide pills are the things that you see in movies where, you know, people have one in their tooth and they bite down on it, and it does cause death fairly rapidly. But if you were to administer thiosulfate, that would stop the behavior. Now, another example of this is heavy metal toxicity. So heavy metal toxicity, these would be things like mercury would be a really good example. So Hg2+ or lead 2+ are examples of heavy metals, and that's just due to their ability to render part of the enzyme ineffective. So these things will render part of the enzyme ineffective. Right? And these metals act by combining with the groups found on many enzymes, like these thiol groups here, right? And so, for example, you can see the thiol groups will bind to the mercury, and then it will inactivate the enzyme. The other thing that these groups will do as well as they'll cause protein denaturation through that binding. And we learned about that in chapter 19. And so both mercury and lead poisoning lead to neurological damages. And can result in death, right? Now, so how do you treat it? Well, heavy metal poisoning is treated by administering chelating agents. So this can be treated by administering chelating agents. Now, what are chelating agents? Well, these are just substances that bind the metals and hold them tightly. One effective antidote is the chelating agent ethylene diamine tetraacetic acid, which is also called EDTA. So this is a chelating agent. Right? And that will chelate all heavy metals except mercury. So it will bind all heavy metals except mercury, right? So if there is heavy metal poisoning like lead, for example, a calcium salt of EDTA is administered intravenously, and in the body what happens is the calcium ions of the salt are displaced by the heavy metal ions. So the heavy metal ions have more binding affinity than the calcium, and that leads to that binding more tightly. And so then the lead EDA complex here, that's soluble. And that's really important because that means that it can be excreted in the urine. And so that is how it's flushed out of the body. Now, the next example that we're going to talk about is penicillin. So kind of the previous two examples we've talked about have been toxic, but not all enzyme inhibitors act as poison. Penicillin is an enzyme inhibitor, right? Right. And sulfa groups are also enzyme inhibitors. So penicillins and sulfa groups are the two really well-known families of antibiotics that inhibit specific enzymes essential to the life processes of bacteria. So these are enzyme inhibitors that inhibit the life process of bacteria. So they're good for us, humans, but they're bad for bacteria because they inhibit those things. So the first penicillin was discovered in 1928 by Alexander Fleming, and he noticed that the bacteria on a culture plate didn't grow in the vicinity of a mold that had contaminated the culture, and that mold was producing penicillin. Now, this is the general penicillin structure here, and his name was Alexander Fleming, and he discovered that in 1928. Okay. Now, the general penicillin structure, right? You'll notice that it has an R group right here. Okay. And what we see is that these then are the different R groups that are attached to that penicillin. And you can see how they're given there. So penicillin will interfere with transpeptidase, which is an enzyme that's important for bacterial cell wall construction. So that inability to form those strong cell walls will prevent the bacteria from surviving, and the bacteria dies, and then you get better. So the last time I was given penicillin, for example, was strep throat. And so strep throat was caused by a bacteria, and penicillin helped get rid of that bacteria. Now, let's talk about reversible inhibitors. So a reversible inhibitor, which is in contrast to those that we previously talked about that are irreversible, reversibly binds to an enzyme, right? And what happens is when it binds to an enzyme, so if we have an enzyme plus an inhibitor, right, and it reversibly binds, we get an equilibrium with this enzyme inhibitor complex. So an equilibrium is established. And so then what that means is the inhibitor can be removed by shifting the equilibrium. So this is based on Le Chatelier's principle, right, where you can increase the concentration of something on one side and it will shift to the other side. Okay. Now, there are two types of reversible inhibitors. There's competitive inhibitors and non-competitive inhibitors. Now, a competitive inhibitor will compete with the substrate for binding at the active site. A non-competitive inhibitor will bind to the enzyme at a location other than the active site. Okay, so competitive inhibitors often have a molecular structure that is similar to the normal substrate of the enzyme. So competitive inhibitors often have a molecular structure similar to the normal substrate of the enzyme. Okay, non-competitive inhibitors generally do not. So let's look at competitive inhibitors in more detail. So a really good example of this is sulfa drugs. So this is an example of a competitive inhibitor. And so, and what sulfa drugs do is they take action on bacteria. So this is sulfa drugs in bacteria. Okay. So what happens is folic acid is a substance that's needed for growth by bacteria, and it's normally synthesized within the bacteria by a chemical process that requires p-aminobenzoic acid, which is shown right here for you. Okay. Now, sulfonamides here, this is the first sulfa drug, resembles p-aminobenzoic acid. So this resembles that. Okay. And so what happens is that sulfonamide will compete with p-aminobenzoic acid for the active site, right, of the bacterial enzyme. Okay, and so when it binds, it can prevent that bacterial growth. So what happens here is we think this is because the enzyme will bind readily to either of these. And so if you introduce a large quantity of this sulfonamide into the patient's body, it causes most of the active sites to be bound to the wrong, from that bacterial point of view, substrate. So instead of binding to the p-aminobenzoic acid, it binds to the sulfonamide. Now, that means that then the synthesis of folic acid is stopped. So the synthesis of folic acid is stopped when sulfonamide is used, right? Which is why we have that X there, okay? Now, so then that prevents the bacteria from multiplying. Right? And then the normal body defenses can really work to destroy them. Now, human beings also require folic acid, but they get that from their diet. And so sulfa drugs then really exert no toxic effects on humans overall. However, people can be allergic to sulfa drugs, and sulfa drugs, particularly if you're given in excessive amounts, can cause intestinal upset because it destroys the intestinal bacteria as well as the harmful bacteria. So sulfa drugs generally have been replaced by other antibiotics like penicillin or tetracyclines, but they're still used in certain bacterial infections because they're just superior, including those in the urinary tract. Now, let's talk more about competitive inhibition. So with competitive inhibition, this is a really beautiful figure for us to look at, to kind of imagine. We have an enzyme, and the substrate should fit in the enzyme, but what happens is an inhibitor comes here and blocks that substrate. Now, all this means is there's competition between the substrate and the inhibitor for that active site. Competitive inhibition can be reversed by increasing the concentration of the substrate. Because we have an equilibrium where the inhibitor's coming in and out and the substrate's coming in and out here, if we just have so much more of the substrate, then the inhibitor is just not going to go in, okay? And so what happens here is if we increase the substrate here, according to Le Chatelier's principle, we're going to increase it here, it's going to shift the equilibrium that way, and it will, in general, end up with more enzyme substrate bindings. Okay, so we increase the concentration of the substrate, and that results in Le Chatelier's principle. Basically working. Now, you can also have non-competitive inhibitors. So non-competitive inhibitors, really, I want you to remember, they really bear no resemblance to the normal enzyme substrate. Right? And they bind reversibly to the surface of the enzyme at a site other than the active site. Right? And the interaction between the enzyme and the non-competitive inhibitor, so we've got an example here where the inhibitor is going to bind over here, not to the active site, but it changes the shape of the active site, for example. So that interaction between the enzyme and the non-competitive inhibitor is going to change the three-dimensional shape of the enzyme, and its active site then will change. And so what that means then is the enzyme is no longer going to bind to the normal substrate, or the substrate will be improperly bound, and that's just going to prevent the catalytic groups of the active site from participating and catalyzing whatever reaction is needed. So when they bind, then what happens is the interaction, let's give some words to this, between the enzyme and the non-competitive inhibitor causes the three-dimensional shape of the enzyme and its active site to change. And when that happens, the enzyme no longer binds to the normal substrate. Now, unlike competitive inhibition, non-competitive inhibition cannot be reversed by the addition of more substrate, because additional substrate doesn't have any effect on enzyme-bound inhibitor, right? It can't displace the inhibitor because it can't bond to the site that is occupied by the inhibitor. So substrate concentration does not affect inhibitor action, right? So that's just a difference there between the competitive inhibition and the non-competitive inhibition. Now, let's talk. So that ends our discussion over inhibitors. And now we're going to kind of switch gears and talk a little bit about regulation of enzyme activities. So enzymes work together in an organized but particularly complicated fashion to facilitate all the biochemical reactions needed in a living organism. Now, for an organism to respond to changing conditions and the cellular needs, you need very sensitive controls over enzyme activity. So we're going to discuss three mechanisms over the next few slides that really exert this control. And those include activation of zymogens, allosteric regulation, and then genetic control. So, a common mechanism for regulating enzyme activity is the synthesis of enzymes in the form of inactive precursors. And these are called zymogens or proenzymes. So these are just inactive precursors of enzymes, right? Now, some enzymes, the reason that this happens is that some enzymes in their active form would degrade the internal structure of the cells and then would degrade the cells themselves, okay? So some enzymes that would degrade the internal structure of the cell are stored as these inactive zymogens. Now, when the active enzyme is needed, the stored zymogen is released from storage and activated at the location of the reaction. Okay, so, and that activation usually involves just a cleavage of one of more of the peptide bonds, right? So a really good example of this is the synthesis of trypsinogen, which is the zymogen. So this here is the zymogen. And I want to remind you, well, trypsinogen will form trypsin. And I want to remind you, we've got some digestive enzymes like pepsin and trypsin and chymotrypsin that digest proteins. So we don't want those to just be in a cell, right? So trypsinogen, for example, is synthesized in the pancreas. So this is synthesized in the pancreas. As this unreactive zymogen. And then it travels to the small intestine where it's activated by hydrolysis. So then it travels to the small intestine where it's activated by hydrolysis. And this is to keep it from just chewing up all of the proteins and things along the way, right? And it's just a hydrolysis of a single peptide bond. And then that active enzyme trypsin is ready to go through and help with digestion. It also forms a hexapeptide as well. Now, and hexa just means 6, so it's like a little 6 amino acid peptide. Now, let's talk about allosteric regulation. So a second method of enzyme regulation involves the combination of an enzyme with some other compound, like a three-dimensional conformation of the enzyme being altered and its catalytic activity is changed. So when we talk about allosteric regulation, this refers to the control of an allosteric enzyme. Okay? And an allosteric enzyme is just an enzyme with a quaternary structure whose activity is changed by the binding of modulators, right? Now, and when we talk about a modulator, this is just a compound. Ooh, I have that definition already for you below. This is just a compound that alters enzymes. Okay, and they bind to the enzyme's active site, and they alter the catalytic ability. So they may increase or decrease that activity. If they increase the activity, they're called activators. And if they decrease the activity, they're called inhibitors. Right? And so non-competitive inhibitors are examples of modulators. So we've already looked at those. Right? Now, enzymes that have that quaternary protein structure with distinctive binding sites for modulators are referred to as those allosteric enzymes, and those are often located at key control points in cellular processes. In general, activators will act as signals for increased production, and inhibitors are used to stop the formation. So an activator is used as a signal to increase production, and then an inhibitor is like, hey, we need to stop production of whatever molecule the body needs, right? So an activator will come along when you need a molecule and say, hey, it's time to make that molecule. And then an inhibitor will come along and say, hey, we've made plenty of that molecule, it's time to stop it, okay? So I kind of view these as like red light, green light for your body. Now, we also have something called feedback inhibition, and this is just a process where the end product of a sequence of enzyme catalyzed reactions inhibit an earlier step in the process. Now, let's look at an example of allosteric regulation. And a really great example of this is an amino acid synthesis of isoleucine. So the synthesis of isoleucine is a 5-step process, and threonine deaminase, which is shown right here, is the enzyme that catalyzes the first step of the conversion from threonine to isoleucine, okay? And what happens here is isoleucine will act as an inhibitor for this process. Now, the structures of isoleucine and threonine are quite different, so isoleucine here is not a competitive inhibitor. So what we find, right, is what happens is when your body needs isoleucine, then this reaction will go on just as it's supposed to go. But when your body has lots of isoleucine, then isoleucine will inhibit the production of isoleucine. So it's an inhibitor right here. And so it'll kind of say, hey, stop, we don't need any more. And then when your isoleucine concentrations go down, then we can start producing again. Now, the site where isoleucine binds to the enzyme is also different from the active site that binds to the threonine here. And that second site is called the allosteric site. Okay, so we talked about that being the allosteric regulation. And that specifically recognizes isoleucine. And isoleucine, when it's there, induces a change in the conformation of the enzyme so that threonine binds really just poorly in that active site. So isoleucine exerts an inhibiting effect on the enzyme activity, and as a result, the reaction slows as the concentration of isoleucine increases, and then you don't get a bunch of excess isoleucine being produced. And then when the concentration of isoleucine falls to a low enough level, the enzymes become active again and more isoleucine is synthesized. Now, this type of allosteric regulation, where the enzyme that catalyzes the first step in a series of reactions is inhibited by the final product, this is an example of feedback inhibition. So this is our example of feedback inhibition, right? And the control of that enzyme activity by the allosteric regulation of key enzymes is an immense benefit to an organism because it allows the concentrations of cellular products to be maintained within very narrow limits. So I think, personally, it's pretty cool. Now, another way that we can regulate, or that our enzymes are regulated, is genetic control. So one way to increase the production from an enzyme-catalyzed reaction is for the cell to increase the number of enzyme molecules present. So enzymes are just proteins, and the synthesis of all proteins, including enzymes, is under genetic control by nucleic acids. Okay, an example is the genetic control of the enzyme activity that involves enzyme induction, which is the synthesis of enzymes in response to the temporary need of the cell. So enzyme induction is just the synthesis of an enzyme in response to a cellular need. Okay. Now, this was first demonstrated in about the 1950s on using E. coli. So beta-galactosidase is an enzyme that's required for the utilization of lactose. So what happens is the beta-galactoside bond is here, and this enzyme, which is shown here, hydrolyzes this. And so that enzyme catalyzes the hydrolysis of these two to D-galactose and D-glucose. Okay. Now, in the absence of lactose in the growth medium, there were very few beta galactoside molecules. However, if E. coli is added to a lactose-containing medium, within minutes, the bacteria begins to produce thousands of molecules of beta-galactosidase. And so that's an example of enzyme induction. If the lactose is removed from the growth medium, the production of beta-galactosidase falls back to a really low level. Okay? So basically, the enzyme only produces, the enzyme is only produced when beta-lactose is present, right? So that biological significance of the genetic control of the enzyme activity is that it allows an organism to adapt to its environmental changes. And that couples genetic control and allosteric regulation of enzyme activity, and basically the cellular processes in the body are under constant control. Now, I know this is a lot as we talk about control, and so I love this kind of summary chart of enzyme activities. So when we look at inhibitors, there's two types of inhibitors. There's irreversible inhibitors, and there's reversible inhibitors. If they're irreversible, they bind tightly to the enzyme and won't let go. And these are like cyanides and heavy metals. If they're reversible inhibitors, they can bind to the active site, and they're competitive inhibitors. And then if they're non-competitive inhibitors, they're going to bind to another site that's not the active site, but will change the shape of the active site. So, as always, if you have any questions, please let me know. This finishes up chapter 20 for us, and we will look at nucleic acids in our next chapter.